It is now 2:01. Hello, everybody. Thank you for coming. My name is Donovan Slubbard. I am a second-year PhD student. I work under Prof. Francesco. He can't be here today, so I'm just going to do the introductions, and then they'll get started with the lecture. So this is for the mini-school in empowering biodiversity, monitoring through data cubes, techniques and applications for open science. There will be 4 of these lectures across of October. Today is the first one that handles over Introduction to Data Cubes, Fundamentals and Applications. The next lecture will be on the 9th of October. Okay, so here I'm just going to read the speakers' biographies quickly. So first up, we have Sandra MacFadyen. She's a research fellow with the Mathematical Biosciences Lab, BioMath, at Stellenbosch University. She earned a joint PhD in botany from Stellenbosch University and landscape ecology from Vrije University Amsterdam in 2018, alongside an MSc in Geographic Information Science and postgraduate degrees in Nature Conservation. Sandra is a core member of the EU-funded Biodiversity Building Blocks for Policy Program, a NITheCS associate, and a principal investigator in a NITheCS research program. Based in the Kruger National Park with the SANParks Savannah Science Unit, she specializes in macro-scale ecosystem dynamics and applied spatial statistics for biodiversity conservation. Through her roles at BioMath and NITheCS, she focuses on advancing novel mathematical models to tackle critical ecological challenges. And the second speaker who will be joining her today is Maarten Trekels. Am I saying that correct? I hope so.
Yes.
He is a biodiversity data scientist and project coordinator at Mesa Botanic Garden, Belgium. With a background in physics and industry experience in aerospace and medical fields, he shifted to biodiversity data through developing standards for the Biodiversity Information Standards Organization under EU-funded projects related to the DISSCO, so DISCO, research infrastructure. He currently coordinates a local implementation of the research infrastructure in Flanders and contributes to FAIR data, FAIR data strategies and interdomain, don't know what that word is, as a member of the Research Data Alliance. Maarten is also a work package leader in the EU-funded Biodiversity Building Blocks for Policy program and is pursuing a PhD at Stellenbosch University, focusing on community ecology. Cool. You guys can get started, introduce yourselves as well if you want, and then I hope you guys enjoy. Thank you so much. That's all from me.
Thanks so much, Donovan. I'm going to kick us off. Welcome everyone to our first session on empowering biodiversity monitoring through data cubes. I hope you can all see my screen. Should be having the empowering biodiversity slide on your screens now. Perfect. As Donovan said, I'm Sandra MacFadyen, and together with Maarten Trekels, we're going to explore with you how data cubes can transform your biodiversity monitoring. We've already had quite an extensive background already, so I'm not going to go through all of this again. Just to say that I'm originally trained as an ecologist, and I've worked for many years with South African national parks before I started to pursue my PhD in later years. But I still live and work in the Kruger National Park, and as such, my interest is in macro-scale ecosystem dynamics and applied spatial temporal statistics for biodiversity conservation. Maarten can tell you more about himself, but he's a biodiversity data scientist at Mesa Botanic Garden, and he's just started a PhD with us at the Department of Mathematical Sciences in Stellenbosch. Maarten?
Thank you very much. I don't think I need to add much more. So although I have a background in physics, I ended up in the biodiversity research through the fact that we are a collection holding institute. So we have a big natural history collection that underwent a digitization program, which generated a lot of data. So I rolled into the field of data science through the digitization projects of our herbarium. Thank you very much.
Great. So over the next few weeks, we aim to introduce you to the idea of data cubes, as a scalable open source solution to address global biodiversity challenges. And today we're going to start off with session 1. It's going to be co-presented by myself and Maarten. Maarten's going to be manning the chat while I speak, and I'll man the chat while he's speaking. And it'll begin with a general introduction to biodiversity and data cubes, and then take you through into the different types of data and the different data standards, and having a look at data publication, which is becoming a big thing now, and the FAIR data principles. And we'll end off with some homework for you to do before the next sessions. So today's one is going to be very introductory. The next 3 sessions are going to be a lot more hands-on. So you'll need to set up a few free accounts so that you can follow along with us. We will try to make it as interactive as possible, depending on the number of participants we have. But otherwise, one of us will be manning the chat while the other is speaking. The second session, on the 9th, we're going to be looking at how do you build data cubes, and Maarten's going to handle that presentation, looking at an introduction to GBIF data. He's going to provide a hands-on guide on biodiversity monitoring through that platform. And then on the 16th, I'm going to take you through a session on hands-on ecological modeling with data cubes. You'll learn how to create your own data cubes through GBIF, and we'll be using Google Colab and Earth Engine so that you can code your own inputs as well. And at the end of that, we will be able to visualize your results on an interactive map. We'll have a look at analyzing the patterns of things like species diversity and relating those back to different environmental variables like climate. We'll end on the 23rd with a session on open science. Maarten and I will co-lead that. And we'll look at how do you share and disseminate your results, with a special focus on using GitHub and Zenodo. And we'll wrap up the mini school after that. And if you can start thinking now already about different topics that you'd like to explore in the next mini school, so that we can keep our mini schools in line with your needs. Okay, so to start off with this lecture today, as I said, it's going to be an introductory lecture. We're going to run through some key principles so that when we start doing some more hands-on coding, we'll all be on the same page. All right, so what is biodiversity? It's otherwise known as biological diversity. It's the variety of all life on Earth, but it also includes all the ways that these living organisms actually interact with each other and the environment. And it gets complicated as you start moving through the different dimensions of biodiversity. And that's why we want to show you how data cubes come into play. So the different scales of biodiversity, it's not just the number of species that you have in a habitat, but it starts off with genetic diversity, looking at the variation in genes within species and allowing for adaptability and resilience when having higher diversity of genes within species. The species diversity, as we all know, the variety of species within an ecosystem or across the planet, depending on your region of interest. And then ecosystem diversity, which is the variety of ecosystems, things like forests or grasslands or even oceans. And they provide different habitats and services. So it's important to capture that diversity as well. So those are the different scales, but we also have different components to biodiversity. And most of the time, when one talks about biodiversity, one often is thinking more along the lines of compositional diversity. So that's the identity and variety of species, genes, or ecosystems in a given area. It includes things like species richness and genetic diversity within species, for example. But there's another component, like structural diversity, which looks at the physical organization or pattern of biodiversity, including the spatial distribution and abundance of species within ecosystems. It's also the arrangements of ecosystems across the landscape. And lastly, we have functional diversity, which looks at ecological processes and the roles that they play in maintaining ecosystem health and services. This includes things like nutrient cycling and energy flow, as well as species interactions like predation or pollination, for example. So to illustrate the different components as scales, we can start off with compositional diversity, which takes us through looking at genes, species and populations, to ecosystems and communities, and ending with landscape types. But each of those have their own scales. Structural diversity, we've got the different levels, as well as functional diversity. So as you can imagine, it gets complicated very quickly. And to add on to that, there are still different ways to measure diversity as well. The most common of which, alpha diversity, which is the variety of species within a single site or ecosystem. This is commonly called species richness, just the number of unique species that you have. Then you've got gamma diversity, which is the total number of species in a site or an ecosystem within the landscape. Then you have beta diversity, which is a turnover in species, so the difference in species composition between two sites or two ecosystems. And lastly, there's a new concept that's zeta diversity, which is essentially the number of species shared across any number of sites or ecosystems. So why is it important? Why should we even care? And all species, plants, animals, depend on each other for survival. That includes us. So the loss of biodiversity leads to the loss of services that nature provides that are essential to the functioning of our society and our economy. The biodiversity loss directly threatens ecosystem health and the services they provide, which is ultimately affecting a risk to human well-being. Because of the importance of biodiversity, the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services was formed for the conservation and sustainable use of biodiversity, long-term human well-being, and sustainable development. And the recent reports that are coming out from this study, it makes it clear that more efforts and comprehensive actions are needed to halt biodiversity declines. There's also the Sustainable Development Goals that were developed in 2015 by the United Nations. And they're part of the 2030 Agenda for Sustainable Development. And they aim to protect biodiversity by promoting sustainable use of ecosystems, conserving species, and ensuring that human activities don't harm natural habitats that are vital for a healthy planet and essentially human well-being. So biodiversity is the foundation of a healthy ecosystem, and we're in the midst of a global biodiversity decline. How can we respond? And data cubes are an excellent tool to be able to grapple with the complexity that we have within biodiversity itself. They're a scalable approach to measuring biodiversity change across components and scales. And throughout this mini school, we're going to explore how cubes can enhance biodiversity monitoring and support global conservation efforts. But before we get there, we first need to just unpack what essentially is a data cube. So I'm going to take you through a few examples specific to biodiversity. All right, so data cubes are multi-dimensional arrays of data that are often used for spatial temporal analysis. They have a definite dimension and structure to their making. Time is an essential component along with space, and then different variables of interest. So, for example, environmental variables that you're interested in. So if we unpack this and have a look at time, for example, a simple graph that shows the changes of rainfall over time can be seen here. And then coupled with that, we can have a map that looks at the long-term mean rainfall, showing the spatial arrangement of rainfall. a lot of these maps will be focused on the Kruger National Park. So if you see that shape, that's Kruger. So a lot of my work is there. But there is still a spatial temporal link that's often ignored, where rainfall is variable not just over space or just over time, but it varies over space and time. And when we're considering things like biodiversity, there are also a number of different variables that interact with each other and the environment that we need to include too. So these can start adding on multiple dimensions to our array. And we mustn't forget scale. So for example, if we look at a spatial scale, is our spatial scale global, regional, local? Is it 100 kilometers, 10 kilometers, 1 kilometer? And those are the type of things that we need to consider. And time as well. There is also a scale to time. Is it 1 hour? Is it 1 day, 1 month, 1 year, or so on? So it's important to choose the right scale for the ecological question of interest and also the development of your data cubes. So identifying the correct variables as well as their interactions is another important factor to consider when we're looking at biodiversity. And by adding more dimensions to our data cube, we can account for these interactions between, for example, genes, species, communities, and their environments, adding this complexity of time and space and linking them, which has previously been hard to do. And data cubes can represent a variable over time and across space. And combining variables into multidimensional matrices, this allows us to also track these interactions and look at the dynamics of environmental change. So to illustrate this in a fun way, here you can see just the spatial temporal variability of things like rainfall, annual temperature, distances to water, dominant ecosystem drivers, or fires. And as you can see, that spatial temporal pattern is there, and that link is there, and that's what we need to try and keep when we start looking at measuring any change. So in other words, if we were to see a pattern in the landscape, it is essentially the response to interactions between a number of different explanatory variables, things like rainfall, temperature, plants, but over time and over space. So how do we visualize this in terms of, in the form of a data cube, for example? If we were to take just one variable, like rainfall in this case, numerically we can start by representing it a single time point as a data frame or a matrix with a latitude and a longitude position. Not sure if you can see my mouse here, but you can consider this a latitude and longitude, and the center point is a grid. Perfect, thank you. So this is just for one time step that we've got here. This is a number of blocks. Remember what I said about scale. We need to think about what is the size of this block. And then these values are the measures of rainfall that we have in there. Now, if we were to add multiple time points to that same latitude and longitude, we're essentially creating this data cubes. So in other words, each dimension represents a new time step, and the cells, the measure of interest at that specific location at certain scale that we've defined. That seems pretty simple. We can do the same for multiple variables by adding different dimensions to our data cube. So we could have temperature, vegetation, herbivores. We could plot them all together. If you can visualize this as dimensions within a dimension, because each of these have their own array as well. And the tricky part comes in when we need to start looking at the interactions between the variables and the outcomes, because we mustn't forget that ecological systems are dynamically complex and adaptive. So understanding these interactions are essential for effective biodiversity conservation. And that's where data cubes come into play, because you can essentially link all of these variables to a driver of interest to see how they interact over time and over space at the same time. So while data cubes offer a scalable approach to track biodiversity and environmental change across these different scales, we need good data. So the whole idea of rubbish in, rubbish out. So it's the quality and the utility of the data that dictates whether the results one can be relied on. And you need that well-structured, standardized data set to be able to have any confidence in the results that you get. So to effectively use data cubes, we need to make sure that the data is fair, and fair being findable, accessible, interoperable, and reusable. So I'm going to hand over to Maarten now, who's actually going to take you through these key concepts around this biodiversity data, including the definitions, data standards, and the importance of making this, making any biodiversity data fair. And then we'll take questions at the end. So I'm going to stop sharing my screen, and Maarten will share his.
Thank you very much. Can you see the presentation?
Yes, you can see it.
Okay, good. Let me see. Okay, so I will focus more on the data aspect around biodiversity, so less the biological background. But there are a few key elements that we will have to cover before we can understand how biodiversity data looks like. First, where is all this data coming from? Where are the sources of biodiversity data? How is this data published? To which data standards or not? And what is fair data publishing? So first, what is biodiversity data? And actually, to understand how biodiversity data looks like, we really have to understand that this data is not coming from a single source. Basically, this data can come from many sources, mainly coming from checklists, citizen science, natural history specimens, ecological surveys, camera trap data, big campaigns of big teams of scientists or single scientists going in the field for a very specific study. So there is not such a thing as a one source of biodiversity information or data. This is coming from many different places, and this can imply that some of your data might show some biases towards one thing or another. There are a few less obvious sources of biodiversity data, and I will not go over all of them, but I will take a few examples out that are maybe not so expected for most people. Biodiversity literature is one of these sources of data. There is an initiative by an NGO called Plaati that is digitizing biodiversity literature and the description of species in literature and making them available digitally. And here you can see an example of such a description of species, where actually you can find way more information than at first you would expect. You could find the scientific name of the species, but you can also find where the type specimen was collected, at which altitude, maybe even location, in which collection these are held, by whom it was collected, and where those specimens are today. So all of this data can be used as an historical source for as an historical data source. Moreover, even in this literature, you can find way more than just what species was where and when. It also contains a whole description of the species, in what kind of environment it lives, what their properties are. So there is a whole big data source available in literature that is being liberated. Another source of biodiversity data is remote sensing. By the big amount of satellite images that we have today, we can learn something about the environment and how vegetations look like at a certain point in time. By including this into the picture, the data source grows bigger and bigger as we speak. And how is this data published? also, this is not really a one single source. You can see here on the right-hand side, this is an image taken by a citizen science project from someone taking an image of a fly in the field and recording it. But you can also have the data portals of natural history collections, like on the top right. Their specimens that contains all this information are digitized and made digitally available through many different kind of platforms that look slightly different, but typically hold the same type of information, like dates and species and locality. Then there are also ecological data, which you can find in other data repositories. So there is a whole bunch of data sources available, and it's not always easy to find your data that you would like to study. Luckily, people are using data aggregators to publish their data, so that you don't have to necessarily go first to these individual sources. The biggest or the well-known data aggregator for biodiversity data is GBIF, the Global Biodiversity Information Facility. This is an infrastructure, an international infrastructure that people can use to publish their data and make it interoperable with all kinds of other data sets. Keeping in mind that these data are, again, an aggregation of many different sources, so they are not coming from one single source and are not all harmonized across how these were measured. But it comes with a lot of advantages. Moreover, your data will get way more visibility by being added into a data aggregator. But these data aggregators also can track how your data is being used and track publications that flow out of the things that you did. So it's actually a way to get credit for collecting your data, because you can trace back where people are using it. And the example that you see here is a study from 2021 where people investigated in what type of studies GBIF mobilized data is used. And you see the typical suspects coming up, like species distribution modeling, taxonomic information, but also some ecology, people and nature, spatial ecology. So this is actually the whole range of fields where you can find your data back. And by the increased visibility of your data, you can see that almost 2 papers a day are published from GBIF mobilized data. So the chances that your data is used in a useful manner increased dramatically. But how do we end up that this works? How do we make sure that our data can be used by others and being interoperable with all other kind of data sources? So like I said, they are not all measured in the same way. They are not all published in the same way, but still we can aggregate them together and make one single data source. And here the fair data principles come to mind. I'm sorry, I'm a bit, I lost my words, sorry. What are the FAIR data principles? FAIR stands for findable, accessible, interoperable, and reusable. The most important thing is that all of these stands for a certain aspect of your data that is important. The one thing I will not cover today is accessibility, because this really mainly deals with how data protocols or how the interaction protocols work. The most well-known example is HTTP or HTTPS. This is the way the internet communicates with each other. So basically, accessibility can be covered by this matter. But we will first start with the findable. And findable, you can see there are a few things. Data or metadata are assigned a globally unique and persistent identifier. You are described with rich metadata, and they are registered or indexed in a searchable resource. So those are the main questions. How do we reach that with our biodiversity data? And actually findability or this persistent identifier, you already know what a persistent identifier is. If you published a paper or you read a paper, you will see that there is a DOI attached to your paper. This DOI is a persistent identifier that always will remain there and will link back to your paper. The data sources or the data aggregator we already mentioned as well are GBIF, and by extension, it was already mentioned in the introduction, DISCO, which is a distributed system of scientific collection, who is trying to unify collection data. In DISCO, there will be also a system of DOIs assigned to each observation or each specimen in a collection. With GBIF, there is not such an identifier for each individual specimen, but when you download the dataset, and we will come back to this in the next lecture, you will see that you can assign a DOI to your dataset, or your dataset will get a DOI once you create a download. Next step is interoperability. How do you make sure that your data can talk to someone else's data? And the big point there is that you have to use data standards. You have to use vocabularies that follow the FAIR principles. And if we look at biodiversity data, there is an organization called the Biodiversity Information Standards Organization that deals with these data standards. This is an organization that builds and curates community-agreed standards to be able to talk about biodiversity data. This is an open organization, so anytime you want to spend on discussing how these data standards have to look like, how you want them to look like, if there are terms missing in the standard, you can follow the different working groups within the organization to help build these standards. The most well-known example of such a data standard is Darwin Core. So if you go to the website of the Darwin Core standards, what you will get there is a whole list of classes, and with these classes, some terminology or some terms attached to them. And each of these terms is described what it is and how it looks like and how you should use it. If we zoom in a little bit on one of the terms, for example, event date, you will see that each of these terms has an identifier, a persistent identifier attached to it, a definition, a clear definition on what this specific term means. What do we want to use? If there is a data value in this field, what is it? And some examples. Of course, Darwin Core is not a standard on its own. It's reusing other data standards where possible, and even tries to include other standards of representing data. For example, in the case of event date, the comment is that the recommended best practice is to use another standard, namely the ISO 8601. And this ISO 8601 is defining how your dates should look like. Mainly that it starts with a year, then a month, then a day, and then how an hour. So basically, Darwin Core itself is not something which is standing on itself. And how do you then make sure that your data is according to this standard? Of course, when you have a database lying around or you have data there, you're not typically recording this immediately in Darwin Core. There are tools to map your data to Darwin Core fields. And GBIF is providing you the integrated publishing toolkits, which allows you to map whatever your fields are called in your dataset to the corresponding Darwin Core term. And then we get to the reusability of data. Reusability means that you allow people to reuse your data. So basically, you need to make sure that that people can read your data. And the last one here, the data meets domain, oh, sorry, that was too soon. Meets domain relevant community standards is one of the important one. But of course, as well, you need to have a license attached to your data, and you need to keep a detailed provenance of your data. I'm not going over all of the different aspects of fair data. There are a lot of sub-principles that you can check out on the website of the Go Fair Foundation. But I will pick out the one on the data license. And this is an important one because this is actually something where you make an agreement with others on how others are able to use your data. And you will often see this kind of symbols popping up. These are typically attached to the Creative Commons licenses. I take the Creative Commons license because they are easy to explain, or at least easier to understand. But there are a whole bunch of other usage licenses flying around. And if you talk about software, you often go to things like an MIT license or GNU licenses. But in this case, we will limit ourselves to Creative Commons licenses. So if you ever see these symbols popping up, it does tell you on how people want you to use their data. And to give an explanation on how this works, licenses typically are predefined, and the CC0 license is actually not really a license, but a waiver. So basically, then you say that your data is public domain and anyone can use it without any restriction. The CC BY license is actually the most open license where you still get credit for what you're supposed to get credit for what you published. It says that people need to attribute the creator of the data under a given in a given way specified by the owner of the data. Then you can go even more restrictive, and the last one is the CC BY NC, which says that it only can be used for non-commercial purposes. This is still an open license, but it does give you some restrictions. So it depends on how you define commercial use, but in some cases this might be problematic, and we will get back to this when people are using public cloud services, for example. This can be considered as commercial use. So data that is published under a CC BY NC license potentially cannot be used in these cloud platforms when you are trying to do some data modeling. So basically, this is a warm welcome for everyone to publish your data as open as possible, to ensure that it can be used by others and that you get the most value of your data. How does this look like on GBIF? This is an example of our herbarium data set, which is on GBIF. This is all the digitized specimens in a Master Botanic Garden. So what you can see here is, for example, all the images that are made available. If specimens were georeferenced, you will get a point on a map so that you can see where they were collected. But you also will see under what license this data was published, what is the DOI or the persistent identifier of this data set, who did the publishing, when was it published, and so on. There is a lot of information that you can find on the data itself that can be used. And GBIF contains a lot of data sets, and in the meantime, if you look only at occurrences, we are over 3 billion occurrences at GBIF at the moment. What's next? So what are the new developments going on and new sources of data? So basically, what we have to do, and this is one of the things I try to advertise here, is we have to encourage people to start to keep on collecting data, find better ways of monitoring and better ways of collecting data that are cheaper and more reliable. People are working on this. Also improve methods of analysis, and this will be subject to our next sessions. And this is also a bit, and this is very recent news because this was published yesterday. There are people thinking of how can we help others to do this publishing in a better way. And so in every year, GBIF is organizing the Ebeneelsen Challenge, which this year was awarded to a program called ChatIPT, which is basically using large language models to help people publish their data. Data publishing can be cumbersome, and not all institutions or researchers have a data team at their disposal to make sure that the data is transformed in a correct way, mapped to the correct terms, and published to a data aggregator or a data portal. The system that won the GBIF Ebeneelsen Challenge this year actually uses the large language models to transform raw data sets into something which is compliant to the FAIR principles and has mapped to the Darwin Core standard. This is also an invitation for all of you. The Ebeneelsen Challenge is basically a competition that is organized yearly. So if you have great ideas on what to do with biodiversity data, how to better collect data, how to do novel things with the data that is flying around, please try out, prepare something and submit it to the challenge. You never know that you win it. Another next thing, it's maybe not a next thing, or a thing that is definitely important in our next sessions, and which is important for biodiversity data. Since it's growing at this very fast pace, and you have this huge amount of additional data sources lying around, such as satellite imagery, it's not always possible anymore to compile or to run your programs on your local computers. That's why it's the next big step is to look into cloud computing for biodiversity data. And there are a lot of cloud computing environments already available that contains a lot of satellite images, such as the Amazon Web Services or Google Earth Engine, or even the Microsoft Planetary Computer, that contain a lot of information on satellite images, but also already contain GBIF data. So also on these cloud platforms, the snapshots of the GBIF dataset are published and do be used for others. Those are also interoperable with our data cubes. So doing big simulations and big calculations becomes way more accessible for people to play around with. What's important here is, and like I said, is at these platforms only data that has that is at maximum CC BY can be made available, because this is considered commercial use of this platform. This so is kind of a limitation of what you can do with them. And then we are actually already at the point if there are any questions for us, and I already saw a couple of hands going up. Maybe I should stop sharing my screen.
We'll leave it there.
Oh, okay.
No, that's fine. That's fine. We can put it back again. I just want to have that last slide. Yeah. Thanks. If anyone has any questions, just put your hand up, and then I'll click the button that says allow to speak.
I think I saw twice a hand going up.
It was a hand, but I think maybe they were trying to do something else. I know every time I want to clap, I put up my hand. So it could be that.
Don't run away yet. We still have a few announcements to make.
Yes. Okay, I've just shared my screen. So yeah, if no one has any questions, this is your homework for next time. So I think from next session, things will be a lot more interactive. So I must just see if we can't figure out a way that we don't have to click on to allow you to talk each time, because if you want to ask something, you want to be able to deal with it as we go along. If it's something that we can deal with in the chat, we will do that. we've got one question that says, are data cubes based on mathematics or statistics? Perhaps I missed it. So neither. It depends on what you do with them. So data cubes themselves are a structure of data that allows you to have these multiple dimensions. Depending on what you do with it, comes in the mathematics or the statistics from it. Maarten, do you want to add anything to that?
No, I think you're right. It's really just a way of representing the data, and we will see in next lectures also a way of already pre-processing some of the data before using it.
Exactly, and I think as Megan put in the chat there, it's a data matrix essentially, but it's a data matrix on steroids. So it's 3D, 4D, 5D, 6D, depending on the different types of variables that you have in there. So if it's one variable, it's 3D, but if you start looking at interactions between variables, it gets multidimensional very quickly.
Yes, and I have to stress that when I was going over the data standards and how biodiversity data look like, I did focus quite a lot on how an occurrence data point looks like. If we are talking about interactions, phenology, and all kind of other dimensions that you can add to your data cube, the picture becomes way more complicated, of course. And maybe one data source is still worth mentioning is GLOBI, the Global Biotic Interactions Database, which is a database which is containing all kind of species interactions. They also use data standards such as the relation ontology to make a relation, to clarify what is the relation between two species.
Right, and we've got two more questions in the chat. One is from Tulia. Wants to know if there's a limit on the number of variables that you include in a data cube, as they need to have the same time, and do they need to have the same time frame? So Tulia, you probably answer this better than anyone else. It's all about computing. So I know with your PhD, the amount of data that you had, it comes down to computing power. So you can add on as many as you want, but when you want to start doing any analysis, it depends on the computing power that you have to be able to do that. And that's when cloud computing plays a role.
Mm-hmm. Yes, that and memory.
Exactly, yeah. And then Caitlin asks if we will be sharing these recordings online. Yes, they will be on the NITheCS website once we're done. And it's a pity you'll miss the third one. That's the fun one because it's mine. We'll be doing the ecological modeling. So yeah, so have a look at the recordings, and I'll actually be in Stellenbosch in October. So if you're around. Okay, so if there's no other questions, if you can see the slide here about things to prepare for next time. So as I've said already in the beginning, we want to try and make it as interactive as possible, and we want to actually work through some real examples. And to do that, we need you to have these different free accounts set up. So the first is GBIF. You can log on there and just create a free account. In most of the exercises we do, I don't think you actually need a GBIF account, but if you want to go on with this and use it for your own data, it's a good idea to have it. And then if you get any problems, you can ask us during the course. And then Earth Engine, we need to set up a free account for that. You need to have a Google account, obviously, for that to work. All of these Google products, same as Google Colab. If you've got a Google account, you just link it to that. I've put a few links here in the slides. They're not the definitive links. I just did a quick search to see what we could use. So just Google and have a look to see if you can find something that tells you how to, if you get stuck. It should be quite straightforward. And then Colab is going to be the medium in which we're going to actually code. So rather than trying to set up an environment on each of your computers, which can be quite problematic, we're going to code online. So you just need to create a free account for that. And we have
Paloma
one question.
has a question. Yeah,
Mm-hmm.
I allowed you to speak, so you can unmute yourself.
Yeah. Paloma, you can unmute yourself and talk.
Oh, sorry, I think I clicked by mistake, so I don't have a question. I'm following.
Okay.
Thank you, Maarten.
No
Sorry.
problem, no problem.
Apologies.
Sure, no worries. Okay, yeah. So if you have any problems setting up these accounts, you can send us an email. I think our contact details are on the NITheCS advert, but it should be pretty straightforward. That's all you need to do is set it up and get an account, and then we're going to take you through each of the steps as far as downloading the data, creating data cubes, and then looking at more detailed coding on how do you do some basic analyses. We'll see how much time we have. If we have spare time, we can always try and troubleshoot someone else's problems. That's always quite fun. So if we have a little extra time, if you've got your own data that you've been struggling with and you want to try and do something similar, we can try and troubleshoot some of that as well.
Yes, and also don't hesitate. So for the people that are already actually using, for example, GBIF data cubes, and I see a few names that might be already using GBIF cubes, that have issues or specific questions for one of the next sessions, don't hesitate to send them in advance. If we have time, we can have a look at it and go over it in the session.
Great. So yeah, I think we're 5 minutes left. But if there's no other questions, you can use this 5 minutes to go and set up your account so that you're all set up for the next one. Yeah, so a student, and I find the infos and questions quite great. Yeah, perfect. All right. Thanks so much, everyone, for coming. We appreciate it. And hopefully we'll see. We're going to keep a list now. You all have to be here next week, too. That'll be the fun stuff. No more lectures, I promise. No more talking at you. We'll be talking with you and trying to get some results. So thank you so much. And see you next week.
See you.
Bye.